Welcome everyone to the last session of the Genome Application Symposium. So this session will be devoted to genomics at the Mediterranean Basin, common questions, challenges, and patterns. And it will be co-chaired by Tereza Manousaki and Maximilian Wagner. So the floor is yours.
Thank you, Jose. Thank you, everyone. So I will share. So can you see my slides?
Yeah.
Great. So I will start with an overview, with a small dive into the Mediterranean Basin. And then Max will give us a very nice case study, and then we'll have our Mentimeter for a discussion. So stay strong until the end. So Mediterranean Sea, that stands for right in the middle of the Earth. It's the largest and deepest closed sea of the planet, and it has so many countries surrounding it. And here I have, through the Wikipedia, I took those countries that have exclusive economic zone. So it's really a sea that has many, many different countries and cultures surrounding it, and very complex. Okay. So about 5.9 million years ago, the Mediterranean Sea almost completely dried out through what we call the Messinian salinity crisis. And this shaped dramatically the biodiversity that we also see today. This finished about 600,000, so up to 5.3 million years before now, to the Sankliad flood when Atlantic waters again entered the region. So the Mediterranean Sea is a biodiversity hotspot. I have here the Posidonian meadows, which are biodiversity hotspots within the Mediterranean. There's a very nice paper by Marta Cole and her colleagues published in 2010, where they describe in detail the species in different taxa that someone can study, can see in the Mediterranean. So there are about 17,000 species, and 20% of them are endemic in the Mediterranean. So we have really iconic species like the monk seal and so on. So it's really extremely rich, and we really have to study it. The biogeographic regions here, you have the different regions, the different smaller seas of the Mediterranean. So there's lots of micro, different smaller regions. You have a temperature gradient, so the eastern side is much warmer compared to the western side. And with the climate change, this is really becoming much higher with the global warming. The production is again higher, is higher on the west side, opposite to the temperature. And you can also hear the different, the depth across the basin. So the distribution of species is, you can see here. So there are many more species at the west side compared to the east side. Same for rayfish and elasmobranchs. But you can also see the endemic fish with some really nice hotspots. And what is really interesting is that there is very high frequency, high number of alien fish at the eastern side, which is of course linked to the source of the alien fish, which is the Suez Canal. But we will talk about that. So what are the major threats of the Mediterranean Sea? It's of course human activities that leads to habitat loss and degradation, extreme pollution. Plastic, of course, is really a huge problem. Overexploitation. So this region is being exploited for many millennia, thousands of years. And of course, right now at the extreme level. natural disasters, invasive species, especially for the last 150 years. And then many different kinds of human disturbance. Max will talk a lot also about that, and bycatches. So you might have reduction in populations of species that are not the target of the fisheries, but are there bycatches. So that's what we have to deal with, to face. I will talk about the source of the invasive species, which is the Suez Canal. It was opened in 1869. It was one of the big changes because it changed the global trade because the ships don't have to go around the whole Africa. So here you can see the most recent opening. So since 1869, there were different openings of the canal. So make it larger and deeper so that more ships can go through it. This is the last one that ended in 2015. From 2015, what I can tell you is that we have many more invasive species. So what happens is that with all those gradual openings, more and more different species can enter the sea. And we can see that. I really like this paper because it had an estimation of the costs in the Mediterranean that is caused by the invasive species, and this has been there's a steep rise the last years. So we have many more invasive species in the last years, and that is due to the opening of the canal. This is one factor, but also the Mediterranean, due to the global warming, it's becoming much more similar to the Red Sea. It's becoming tropicalized, so it's warmer, and so the species that are from the Red Sea are just they're happy also in the Mediterranean. So they are invading. They have no competitors, no predators. Most of the times. One of the most notorious fish species here, especially in Greece and in the whole Eastern Mediterranean, is Lagocephalus scleratus. It's a puffer fish. It's poisonous, so we had to really warn that they should not be consumed. And it's really taking over. It has reached the Italian waters, and it's really a burden for fishermen also, even for the tourism. There, in the news, there were sections saying that they actually had attacked tourists, which was not exactly the case. So we have the invasive species in the Mediterranean. If someone is studying populations and does genomics structure analysis in the Mediterranean, they will see the biogeographic barriers that have been very nicely explained in the paper by Heras et al. 2019. So there are so you have from west to east the Strait of Gibraltar, obviously, but then there's also the Almeria-Oran front, which is where actually the Atlantic water and the Mediterranean water are borders. So if you study population here and you do a genetic study analysis, most of the times it will cluster with the Atlantic population. And here we have the West Mediterranean populations. We have the Ibiza Channel, and then the Balearic front, the Strait of Sicily, the Suez Canal, and then in the Red Sea, we have the Strait of Bab el-Mandeb. I found this picture really interesting from Seyfried et al. Again, 2019, because they combine the landscape. They also have the main wind currents of Tramontana and Mistral, and they also show the different currents of the region. This was a really comprehensive view of what is happening and what we need to take into consideration when we're studying population structure in a region. And with that, I will leave the floor to Max, where he will give a case study.
Right, let me quickly share my screen. Right.
Yeah.
All right, yeah. For me to give a short talk about the case study that I'm currently working on. And thanks, Tereza, for the nice overview of the factors shaping Mediterranean biodiversity, because it's also very relevant to the study system I'm mainly focusing on. And I've started like working on gravel beaches already during my bachelor's, and that's when I kind of got into also my PhD, and now I'm a postdoc at the University of Rijeka, and still working on gravel beaches and ecology. So when researching some years ago what kind of brings people to Croatia, there were some funny, like there was a poll by the Telegraph and some funny answers. But I think the most impressive answer was, or what most people liked in Croatia, are the spectacular gravel beaches that they have. I mean, nothing compared to Greece. Greece has even more nicer beaches and even more gravel beaches. But you have this certain kind of special appeals, like the one here, the Zlatni Rat, the Golden Horn on the island of Brač, which is very, very astonishing. So let's talk about another factor that's not really temporarily shaping, like in the past, shaping the diversity that we see now, but more at the moment. And that's, of course, tourism. Croatia and also Greece, as you can see here, unfortunately, this slide is only in German. But what it basically shows is that Croatia and Greece has a very high GDP in tourism, and it's an older study, so it might be even higher by now. And also the number of visitors, when we specifically look to Croatia, increased starting from 2016. So tourism became quite popular in the region. And with the tourism, also people started restructuring and building beaches. So just to give you this slide on the top, on the bottom here, that shows the amount of nourished or supplied beach sediments in Croatia. And as you can see, with from 2018 onwards, the amounts have been almost quadrupled at some places. So that means there is a strong correlation of building new beach areas, and that correlates with tourism. And of course, beaches need to be managed somehow, especially in urbanized areas, because beaches will just not stay stable, they will degrade, they will erode, and beaches are very important for avoiding coastal erosion. So they have some certain also need to be maintained and managed. But of course, this dramatic increase in new beaches and very often poorly designed beaches was quite frightening, and especially frightening for some of the animals that live there. And this is also the study organism that I mainly focus on, the Mediterranean endemic genus Guania. So it's a purely endemic fish that lives right underneath the feet of tourists, basically. So everybody who has been to any Mediterranean gravel beach likely stepped on these little guys. They are really there in numerous numbers, like up to 50 individuals. And like we've learned before, they follow nicely the pattern of Mediterranean biodiversity. They evolved likely after the Messinian salinity crisis and radiated there. And during my PhD, I was already studying this fish, describing some new species. And we found that there are two ecotypes, slender and stout. I will come to that later. And we were just looking into parallel patterns. And we sequenced the first reference genome of one of the clingfishes within the Vertebrate Genomes Project, and with the main goal to detect what is the genetic basis for these parallel patterns. And we did this for one of the Adriatic species, Guania adriatica. Yeah, we found some nice regions and so on and so forth. But what was really interesting was to see in sympatry that these two morphotypes, these two ecotypes, as we found out later, they really separate according to certain microhabitats. So the more slender morph occurs in more narrow gravel types compared to the stout morph that use more coarse gravel. And one side effect of this whole study was when we looked at one of the artificial beaches in Rijeka that you see here, and you can see the sediment distribution there with the fraction-free kind of intermediate size gravel, which is really dominant there, whereas on the right side here is a more natural distribution of sediments. So natural beaches have more like a uniform distribution of sediment types, but the fish still occurred in these types of sediments. So we kind of came up with the hypothesis, maybe these fish, they could serve as nice bioindicators for assessing the quality of already established gravel beaches, or maybe if we design them in the future, what could a gravel beach look like? How could we design them to make them more like sustainable and more natural? We already heard that the Mediterranean is very complex in terms of its seascape. So, and very often, like when we talk about population genetics, we draw conclusions over the seascape towards what we see in the genetic data. And what we've seen also today, like even though large pelagic fishes usually have these good dispersal abilities and they overcome these obstacles, but sometimes they do not, as we know now from the herring of the Baltic Sea, where you have some special cases as well. And the small benthic fishes that I mainly focus on, they really don't get out there. So they show very, very strong patterns, strong geographical patterns, because they have very short pelagic larval phases. They cannot swim in free water. And even the larvae, like they were really well developed already while hatching, and they will rather stay where they are and not go out in these strong gyres. And basically, that's what we see also when we look at mitochondrial CO1 data. We see that we have strong structures for each of the species, but also we see these effects of demographic effects. For instance, in the Guania adriatica, you see this star-like network, which clearly is a sign for population expansion. And that likely correlates with the last glacial maximum when the northern part of the Adriatic completely fell dry. So we get some kind of spatial resolution, especially in the older or more stable environments, like the Eastern Mediterranean populations that you see below. This is Guania hofrichteri and orientalis that show very strong geographic structure, but not so much in the Adriatic ones, probably due to this demography that they went to, this bottleneck they went through. So the main idea is now to study this kind of population connectivity, but more on a very small spatial context in the Bay of Rijeka. Why the Bay of Rijeka? Because it's full of artificially nourished beaches. So it's artificially built beaches. And we want to see where these larvae come from and how they are recruiting and how they are connected. And what we also have is knowledge about certain events that happened in the last years that lead to the building and the restructuring of the beaches. So like these oil spills that we have here. But in order to do so, and we want to do this, and we want to compare these two species that occur both on these gravel beaches, we have to sequence another reference genome. So we now have this reference genome of Guania adriatica, that's the lower one. But we also want to sequence now one for the Guania pigra. And that will hopefully give us the genomic resolution we need to really draw conclusions over a very small scale. And I'm really much aware that this is quite risky and it needs a proper sampling design. So we are still now collecting specimens from several populations, and hopefully we can draw conclusions from where these animals came from and how they recruit into newly established beach areas, because this could also give us nice insights into how fast can newly established beaches be recruited by these top predators, because there are no other top predators in this gravel environment than these blunt-snouted clingfishes. And maybe one side note, because we sequenced this genome within the VGP, the Vertebrate Genomes Project, and that just want to highlight that at the same time where the genome wasn't even published, some people used it and they found this amazing genomic or the lack of genomic signal for immune globulin genes. These fishes lack immune globulin genes and developed a completely different immune system strategy. And I just want to highlight this, that it's really cool to, like, as soon as we have this data available, and we never know, we sequenced this fish from the beach and didn't know nothing, more or less, and then people can take it and find out this amazing stuff. So that's what I wanted to share. And I think we can also go now to the Mentimeter, unless should we also, Tereza, does request for questions considering the talks?
Yes, if there's people who would like to ask something or make a comment. Okay, we can go to the Mentimeter, I guess.
Rob.
Rob.
Yeah, just a quick question about the species complex stuff, and like how much morphology, like observations, measurements, etc., are actually needed to before you get into the molecular stuff to have an idea of the differences, and are there still differences that you think are yet to be uncovered as you examine more populations?
You mean now with my stuff? Considering my, yes. So yeah, so we did it the lazy way, so to say. When I started out, I collected the fish, did some barcoding, and then the differences were clear. And then we looked at them again and found also very clear morphological differences. So for the species descriptions, this is basically classical, we used a very classical approach, just make linear measurements, but also before that, we made 2D geometric morphometrics of the overall body shapes. So to quantify kind of shape differences between individuals, and they already clearly separated, but only between the two morphotypes and not between they are very similar. So they really converged into these slender and stout morphotypes.
Yeah.
And considering uncovering more biodiversity, I'm quite sure there could be more. And I think that's also one of the challenges in the Mediterranean, since it's very small. There are many countries there. To really get the full range, geographic range covered is for sure one of the major complexes within this study. And I tried to reach out, especially to researchers from the North African coast. So far, it was always ending up somewhere, unfortunately. So we definitely have potential, and I still try to get more people involved, and because I'm sure there is way more going on.
Good luck.
Thank you. So
okay, now you need to open the Mentimeter and share.
Sorry, wrong Mentimeter. Where are you? Is this one the Mentimeter? I guess it is.
Yes. Now you present.
No, is it the Mentimeter?
Yes, we do. You need to present now.
Because I cannot see it. It's behind. It's hidden.
At the top right corner, you have the present button.
Okay. Okay. Yes, sorry.
We are learning the tool.
Okay.
So,
all right.
Did the people keep the code? Maybe we can share the code in the chat.
Just give me a second. I need to come out again. Sorry. Wanted to copy it before.
Yes.
That.
Thank you, Jose.
So this is the Menti code. Am I still sharing? Yeah.
You are.
but I'm not in the.
Yes.
Okay.
Perfect.
Yeah. All right.
So we already have answers. Okay, great.
So Teresa, would you want to?
Yeah, so the first question was, what group of organisms are you working on? Hoping to find some marine, yeah, corals.
Nematodes,
You can also
sharks.
scroll down. Yes, we have 2 people working with sharks. We have fish.
Nice.
Yeah. Okay. Fish, sharks, corals.
So we
And
made
some sponges.
All right.
Okay, so then the next question is, what are you trying to answer on those species? We thought of putting the scroll a number of options here, but we said that it's better to keep it open because we cannot predict all the different types of questions. But I guess population genetics would be the most frequent answer. So population adaptation, genomics.
Nice. Demographic changes also.
Quantitative genetics. Okay. Yeah, it's very persistent pattern.
So should we go next to the next question, or you want to discuss
Wait
something?
a bit because there are more people. I see the
Yeah,
responses
sorry, yeah.
still, so
So let's wait a little bit. Yeah.
bit. It would be interesting to see this question in the moment where we had 150 people who we are, but it's a very general question, or not only for the marine species. Okay, let's see then. Okay, so what is the genetic resolution that you have targeted for until now? So I guess this is also a question that has been asked many different Mentimeters. I would say, given you're working on marine species, let's see.
It's funny, yeah.
Yeah.
Live update.
What I find interesting is that microsatellites persist. We were saying yesterday that they're mostly dead, but they're not. They are being used, and they are very informative.
I think many of these Mediterranean population connectivity studies on small fish and stuff that I usually study is based on microsatellite data still. So it's quite informative, and often the patterns are just like elevated using more
Yes.
like whole genome data and stuff. Yeah. but the audience might also be biased here and
so.
spoiled with whole genome resequencing, because I think our quick research also revealed that there is actually, for the Mediterranean itself, there's just, of course, like for any other region in the world, only few reference genomes, or anyway, genomes in higher quality available. Of course, good to see.
So I wanted to say that in BT, there are many marine species in the flow, so there should be many more coming.
Mm. Also, a thing we have not really targeted yet are also, there's of course huge freshwater biodiversity as well. And there is also case studies going on Mediterranean endemic gobies, for instance, also in collaboration from Unizagreb and Greek colleagues. So more genomes on the way.
Okay. So do you have a reference genome for your focal species? Let's see who will win. No is winning, okay.
50-50.
Yeah, the target is that this will change soon, but most of the species will have a reference genome. Let's see how much time it will take to see this. Okay, next question. So if you're working in the Mediterranean Sea, which is the main study area? Which part of the sea are you working on? We have North African coastline. I was expecting the Western Basin to go higher, but it's really interesting with the Adriatic Sea that has such a high endemism, so many endemic species.
Yeah, I think the endemism in the fish slides that you showed is mainly related to brackish and freshwater individuals. So purely marine, there is, to the best that I know, it's only one species, and that's the one that the clingfish basically, where we now sequencing the reference genome, this Guania pigra, and it's to date the only purely marine endemic Adriatic fish, because most Adriatic fishes also occur towards the Ionian Sea, but marine fishes, sorry. But there's definitely potential.
Jean-Baptiste?
Yes.
Hello. Yeah.
Mm.
Yeah, maybe the Adriatic Sea, the main characteristic is mostly on the demographic history rather than the level of endemism. At least for marine species. I guess you have this huge recolonization. after the glaciation, and here you have some imprint of demographic bottleneck in many species. So I don't know if we have to expect such high level of endemism in this sea, but for sure this demographic imprint is already observed in various species, from fish to also in corals. And just a comment.
Yes, good.
Thank you.
Thank you.
Okay, let's move to the next. Okay, then we would like to see if invasive species impact your focal species, if you see interactions. Yes and no. So from those who said yes, what would be one observation that you have? Yes, Jean-Baptiste.
Yeah, so to be sincere, I'm not sure it's really invasive. But yeah, we noticed on Paramecia clavata, on some of this red gorgonian population in summer, in addition to the increase in water temperature, you have a bloom of filamentous algae, and it's like crazy. And if it's lasts too long, the gorgonians are dying. You had some big event already in Catalonia. It should be like in 2021 or 22, something like that. Yeah. It looked like a landscape of Lords of the Ring, you know, like everything was so, I don't know, it was really strange. But yeah, the corals population suffer a lot from this invasive or explosive species.
So we observe new species every, I don't know, some years you see big changes. Like we had the lionfish now for the last, I don't know, 10 years, and they are everywhere. But what is interesting is that we see fish that invaded like in the 1950s, and suddenly their population has grown like crazy. So it's not only new species, but it's also the populations of the already established species that are going up, so, which I guess is linked to the climate change. So these are the things we would like to study at the genomic level. Should we move to the next one? And this is an open question about how genomics can actually contribute to the management of the invasive species, because this is something very hard to answer. This is why we put this question here, because we can imagine that we can really monitor what is happening, but how can we really contribute to stopping the invasive species? If you have ideas. This is towards the direction also of how can genomics really be applied. Just say, no, I'm sorry. I thought you had a question. Early warning, yeah, that's true. So we can study the demographic dynamics. We can do comparative genomics. We can see which are, we can see the adaptive potential. We can see exactly, we can observe the process of the adaptation of the invasive populations to the new environment. We can understand if you have one case of invasion, if multiple invasion events have taken place, if it was only one, like a founder effect. We can compare with the source populations. But how can we go to the management authorities and say something useful to them? That's my main question here. Yeah, early detection, I think, is something that can is the most applied part of that. Okay, let's move to the next. At the same spirit, if you can see the impact of the climate change on the species you're studying.
I wonder, this person who wrote, maybe in the future, where this target species is it more in the Adriatic or?
So everyone, almost everyone is on the positive side.
Mm.
It couldn't be otherwise. I think it would be really important to note down all the different observations and on different taxa that climate change has. It would be really nice for raising the awareness that we all see it. This is really, really important.
Yeah, and especially the Mediterranean basin is really affected strongly,
Yeah.
like on a global scale as well. It heats up way faster, the whole area.
And I have to say that even the taxi drivers, if you enter taxis, you will hear the taxi drivers talking about the climate change, that they can feel it.
Mm.
So it's in everyday life here.
Yeah.
Without, yeah.
We have time for one big last question, last 2 minutes.
Alessia Kagliani is saying also that Adriatic in particular, since it is one of the last cold areas during the winter. Yeah, and we have this question now, and which is, it incorporates all the complexity of the Mediterranean region, East, West, Europe, Africa. We have even war in some places, so it's really complicated place if you're working on species and you would like to sample across the whole distribution. Yeah. Training and sharing of expertise. This links to the question that Jose had about the widening countries, because many of them are in the Mediterranean, which ended up in the collaboration. So this is our last question, right?
Not sure. I think we have one.
Yes.
Yes, we have two more, but I think
Two
we
more.
are almost. I think it's we have 45 minutes, right, for the session.
Yes.
So I think maybe are the answers still there? Yes, maybe. Yeah, maybe stay. I don't know. You should say, Teresa, what you think. Like, should we go for one more or?
Let's move to the next one.
Yeah.
Yes, because the one with the stakeholders.
Mhm.
So how do you engage stakeholders across the Mediterranean, in the studies across the Mediterranean? And if we continue with zero responses, then we have to close the Mentimeter because it's the final part of the whole symposium.
Yeah.
Yeah, I think we can close that because we had a whole session on the stakeholders. So if there are any other comments on our session? Then we thank you for being so strong to stay that late after a second day. Jose, we hand over to you.
Yes, thank you, Tereza and Max, for driving this last session of the Genome Application Symposium, and thank you everyone for staying for the last session. So we are closing the Genome Application Symposium, so I would like to thank all chairs and speakers for driving all the interesting discussions that we had yesterday and today. And of course, everyone that participated in all sessions and participating in the polls, in the discussions. I think it was very insightful, very interesting, and it clearly demonstrated what we are doing to actually apply genomic research to biodiversity conservation, but also other aspects of ecosystem services. So thank you all, and see you next time.